Okay, so as I told you in the beginning, I'm going to be focusing on a very classical technique, which is the processing of tissues and cells and also organelles for studying under the transmission electron microscope. So I'm going to go step by step through different protocols. One of them is going to be to prepare or processing some samples like I told you. We can embed and section tissues, cells, or even organelles, isolated organelles, and we can look at them at the electron microscope. And the first protocol I'm going to be talking about is just to process for conventional transmission electron microscope, which are these type of images that probably you are very used to look at them. This is, for example, this is a swan cell, and and you have also some small axons around it. So it's surrounding some of those axons. So there is also another type of electron of processing for electron microscopy, but it's this way you can immunolabel some proteins, and we're gonna be go through some of these protocols, which are pre-embedding and post-embedding tissues and cells, to be able to localize precisely proteins in these organelles. And then at the end, I'm going to be giving you some applications, ideas, and examples from some of the projects I've been involved in, and just to give you an idea what you can do with all these protocols. Okay. So the first thing is just to understand why we want to use electron microscopy, and it's the main reason is because we want to look at specific molecules or organelles or contents of those organelles at a very high resolving power. So as you see here in this table, the electron microscope is the one that is going to give you the highest resolving power. And what is the resolving power of the resolution? Sorry, of the resolution of a microscope or any equipment. Which the resolving power is the minimum distance that 2 points can be from each other to be discerned as such. For example, you have these purple dots. So something that is unresolved. I don't know if you can see my pointer. Can you see it?
I can. Yes. Yes. We can.
Okay. I don't know if I can use a different one, maybe. Yeah, probably this one is better, right?
Yes.
Okay, perfect. So when there are two points that are too close together, then you cannot resolve them. So these two points on the left side, they are fully resolved because we can see one from each other. So for example, in the electron microscope, in the transmission electron microscope, the resolution power is 0.2 nanometers. So that means that the minimum distance that 2 points can be together, so we can see them as different points, is 0.2 nanometers, which is actually pretty good. So we can see even molecules at some point. Okay, so here there are two examples of the different microscopes that we can use. The one on the left is the transmission electron microscope. I'm going to be talking mostly about this one, but also there is another one, which is the scanning electron microscope, which is with this one we can even see cells or tissues in 3D with a 3D reconstruction. These two microscopes that you are looking at are actually in the University of Salamanca in the facility which is called Nucleus. So, yeah. So the transmission, how does it work, the transmission electron microscope? We're going to go just very briefly through the basic mechanism so you can understand why we need to process the tissue in a certain way. So as you see here on the left, there's a scheme with the it's a cut of the microscope, so you can see what is inside. And up there you have the filament, which is actually the electron source, and then you see the beam, which is in yellow, and that comes through all the different parts of the microscope that's running to the bottom, and then it's going to cross the specimen, which is right here in black. And I'm going to tell you how to process the specimen so we can see what's in the cells. Okay, so here you have the samples holder, and then there's an imaging system at the bottom. Here is actually it's very simple described. Is a lot more complicated than this, but I mean you can have an idea of how it works. So this microscope, what it does, it directs a beam, this electron beam, through the specimen, and then we can image the specimen at the bottom of the microscope. So what it's going to do is to create an image which is based on the electrons that are going to be absorbed at the specimen. And now I'm going to tell you how to process this specimen so we can detect the different parts, which is dark and white, of that image that I showed you in the beginning of the presentation. And then the image is actually focused at the bottom using electromagnets. We are not going to use lenses as we use with optical microscopes, because there's no sense for that, because we are using electron instead of light, instead of photons, I mean. And also I put here an extra tip, because there are extra equipments attached to this microscope, because we need that in this big column where we have the electron beam, and there will be no air at all. So it's airless. So we need vacuum pumps to achieve the level of vacuum that we need inside this container, this column, to produce a consistent electron beam. So we need to actually control where the electron beam is going through. So we need this column to be airless, completely airless, a vacuum, completely vacuum. So here's a comparison of the three main microscopes I've been talking about. This is the light microscope one that you already know. This is the transmission electron microscope, that is the one that we are going to be focusing in. And then the scanning electron microscope on your right. So as you see, we have, I mean, the main components are more or less the same, but they are completely different, because here we have the light source, which is a lamp. But then in the electron microscope, we're going to have an electron source, of course. And then we have lenses like condenser, objective, and also an eyepiece here that we don't have in the electron microscopes, because we don't see electrons. We just see the light. And then you see here you have the specimen tissue section for the light microscope, and also for the transmission electron microscope. But for the scanning electron microscope, we can use like the 3D specimen, like I don't know, like a head or a head of a fly or something like that you probably have seen, this 3D reconstruction of different things. Or for example, here we have a cell with bacteria on top, so we can see a 3D reconstruction of any specimen. Okay, so this works completely different. But in these cases are very similar, the light microscope and the transmission electron microscope. The difference is that we are using electrons to achieve the resolution that we want to look for. I don't know if you have any questions so far, but anyhow, we can ask me later on. You can just write down your questions and we can solve them later. So just to have an overview of what I'm talking about, how this electron microscope is working, is that you have up here the electron gun. And remember that to be able to function properly, there has to be a vacuum in the main column. So the pumps are required to be removing the air from the entire interior of the column. Then we have a condenser lens, an objective, intermediate lens. You see, we have many different lenses, but the difference is that they are electromagnets. They are not glass lenses at all. We have the specimen right here in the middle, as you can see. And then the image is projected based on the electrons that are absorbed by the metals. Now I'm giving you some clues of what we need to have this image properly, that we're going to go through the protocol. Okay, so we need that there could be some metals that are deposited on the sample, and those metals are going to be absorbing some of the electrons, and some others are going to be passing through the samples. So we need to have some contrast in your sample, in your sections, to be able to see something. Okay, for example, like this, we have some parts which are dark, which is the part where the metals are being deposited, and some other parts which are lighter. That depends on how many metals have been, the amount of metals that have been deposited in the different areas of the cell. So we have more or less electron dense extractors. So more electron dense and less electron dense structures in the different tissues, cells, organelles, whatever we are looking at, okay? So now we're gonna go through the detailed protocol for the like the major technique, which is the conventional EM processing of any sample. So I'm gonna be focusing on neuroscience, like in brain, retina, things like that. But this protocol can be applied to any kind of sample that you want to look at the electron microscope. So first of all, we need to do a chemical fixation, and is crucial to use a glutaraldehyde, which is the aldehyde proper developed for this kind of technique. And then we're gonna have to use some of those metals that need to be deposited in the tissue, which is the osmium treatment. Don't worry, because we're gonna go step by step through all these procedures, okay? So this is actually to fix and to stain the tissue. So this osmium is gonna be deposited in certain parts of the cells, which is gonna be the part which is gonna be absorbing the electron beam. And then we're gonna dehydrate the tissue, and we're gonna embed in hydrophobic resin. And this is very important, hydrophobic resin. Remember that I told you that we need a vacuum inside the column. So any rest of water in this tissue is gonna destroy your sample or even damage the column of the electron microscope. So this is very important. We need to dehydrate completely the tissue and embed in a resin. I'm going to be talking about which kind of resins we are using, which are going to be polymerized and hardened. It's going to get very hard, so we can get very, very thin sections. And then we're going to stain and go to the EM to observation. So as I told you, I'm going to be going step by step. I know this is in Spanish, but it's exactly the same as I just told you. We're going to go through all these steps very slowly, so you can understand why we are doing all this processing. Okay, so here we have our sample, which has been fixed. And important to be with a mixture of paraformaldehyde, but also the most important thing is that you have glutaraldehyde in your fixing liquid. And then this tissue can be fixed by perfusion or by immersion. That means that you can perfuse the liquid, the fixative, into the animal, or you can just get a piece of tissue and put it in the liquid, in the fixative, just to fix. Okay. And then we're gonna wash after overnight. We're gonna wash and remove the extra fixative from the tissue, so it's a tampon. And then you go to the second fixation, which is actually to deposit this metal, the osmium, into the tissue. So it's osmium tetroxide. I'm going to be talking about this later. And then after the osmium tetroxide, you go to all this dehydration. You go in ethanol from 70, 90, 96 until pure ethanol, 100% ethanol. It's very important that you dehydrate your samples very well. And after this, we go to this embedment, which is also called infiltration. And then you need what is one other solution, which is called propylene oxide. This is very necessary because I'm going to show you now that this resin is actually a kind of honey-like structure. So it's not completely liquid, it's a little bit thick. So in order to get this resin inside your tissue, you're gonna need this propylene oxide to infiltrate your resin into the tissue, okay? So there's gonna be between 10 and 20 hours until you get a proper infiltration of the resin into your tissue. So then after all these passages, you go and take a mold and pour some of the resin in the mold, put your block of tissue or your sample, your cells, isolated organelles, whatever you want to look at, and put it properly in this mold. This has to be polymerized in an oven at 60 degrees Celsius. Here it says 48 hours, but it's usually with an overnight curing should be enough. And then this block is already hard and ready for sectioning. Okay, So as I told you, I'm going to be telling you a little bit more about some of these reactives and products that we are using for all this processing. One of them is very important, is the osmium tetroxide, which is kind of toxic. All of these resins and reactives are kind of toxic. That's why I'm using here this call. So the osmium tetroxide is a heavy metal and is actually coming here in these kind of samples in vials that you have to break inside the fume hood and then dissolve in water, okay? And with a lot of care, because you cannot touch this. Also because this, even the fumes from the osmium tetroxide can be very dangerous. So and then the plastic resins, you really, I mean, depending on the tissue you want to section, you have to make different mixtures. But that is actually in the manufacturer. So when you get the resin, they tell you exactly the mixture you have to do, you have to get in order to get the proper one for your tissue. Okay. And then once you have done this process, the resin is pure in the mold, and then you put it in the oven. The result, it looks like this. These are actually pieces of ice, which are the examples that I'm using most of the time, mouse eyes, embedded in this mold. And as you see, it's a very strong and polymerized resin or plastic. Actually, it's a plastic. Okay. So once you have done all this process, the fixation, osmium tetroxide, dehydration, embedded in plastic, and then in the oven already cured the resin, and then get your blocks already done and ready for sectioning, you have to go and get these sections done. Something very, very important in this is that you really need to get very thin sections. They are called ultra thin sections. And to get that, you need the ultra microton, is what I call. Actually, it's like a regular microton for any other tissue processing. But in this case, it precisely gets sections as thin as nanometers. And I'm going to be telling you later why we need ultra thin sections or so thin sections. But right now, I'm going to just show you how is the ultra microton. As you see, it's a regular microton. We have here our block in this piece of the machine, of the equipment, and then at the bottom down here we have a knife, and here you can see that this knife is made of glass. It's a glass knife, but these kind of knives are not used anymore. I'm hearing some of a sound, and I don't know if you want to ask me something or there's something wrong. Is it okay? I was listening some noise. I'm not sure. Okay.
This kind of
We didn't mute the microphone, but I think now it's fine.
Okay. I thought that you were asking me something and I couldn't hear you. Okay. So as you see here, we have the block, the resin block with your sample, and then the knife, which here is, I'm showing you a glass knife, but these are kind of old. We don't use these anymore. And if we use them, it's just to trim the block so we can access to actually where the sample is or where our tissue is or whatever, just to trim and make sure that we are getting the right sections to observe in the electron microscope. So actually, I have a couple of videos here just to show you the actual settings that I'm using in my lab. And these are videos that I took. And on the left side, you see that there's a water container and then the knife. This knife is not a glass knife anymore. This is a diamond knife, which is much tougher, and I can get a lot of sections during my entire life. And as you see, here is the water container. The diamond knife, sorry, is here at the very tip. And now I'm gonna put I hope that the video plays, and you can see how I get the sections. Okay, yeah, it's playing. Can you see the video? Playing?
Yes.
Great. Okay, so you see that the block is passing through the knife, and I'm going to turn on the other video so you can see through the eyepiece. And and those are the thin sections. So you see they have like a kind of a yellowish color, more a gold color, and that's because they are really thin. I don't know if you can see there's some other like blue or a little bit darker colors over there, and those are thicker sections. How do I know the sections that I need to look at the electron microscope? Well, here's again the same picture again with the water tray and then the knife over there, and there's some sections floating on top of the water. Okay, so to make sure that I'm getting from here, from this water tray, and the sections that are really good for looking at the electron microscope, I need to go to this chart, because actually the sections need to be around 60 to 90 nanometers thick, from silver to gold. So the previous one that I was showing you, they're actually pretty good to look at the EM level. Why this color? Why this thickness? Well, the electrons that we are using for in this microscope are really good for passing through the sections, but the sections need to be of a certain thickness, because they are not strong enough to cross over thick sections. So we need to get very thin sections in order to get the electrons passing through our section. Okay, so after we choose the sections that we want to look at the EM level, again, and I repeat, they have to be ultra thin sections from 60 to 90 nanometers thickness. I'm gonna use these kind of holders. So in the light microscope, we are using glass holders to put our sections on, because the light can pass through the glass and pass through our samples. But in this case, we are going to need metal holders, and they are like this, are actually very, very small, like more or less like 3 millimeters wide, these holders. And actually you see that they are grids. They have some bars crossing over. Some of them they have this aspect like this, some other kind of grids, different sizes, so on, depending on the sections I want to look at. But I prefer this one that you have on the bottom, because as you see that I have just one hole, and I can actually see my entire sample in just one look. But the thing is that since this is one hole, I need to use like support membrane that I have to make by my own, to make sure that I don't lose my samples through the hole. Okay, so I need some membrane of support to get my sample on it. Okay, so the holders for the electron microscope are these kind of grids. There are many different sizes, and not sizes because they are all the same size. They are all 3 millimeters. Because the holder of the electron microscope is that wide, there's no more. But there are different types of grids, as you see here. There are just some examples. Okay. And now we're gonna get our grid, our sample that we already got in our grid, into the microscope. So here you have the holder, the specimen holder that we're gonna introduce in the microscope. As you see, we have a handle here, we have an O-ring, and we have just at the end, at the edge of this handle, we have these two little holes, actually, where we're gonna put our grid. As you see, it's very precise. You need to have a very nice pose just to put it over there and don't fly away. And so it needs to stay right here. As actually you have a flange here, like a locking, that you just put the locking on top of the grid, so it stays put while you are introducing this holder into the electron microscope. Okay, so as far as we have gone, we have process our tissue for the conventional EM. Okay, so we have done the chemical fixation, osmium treatment. We need to deposit this metal, so we are going to be able to see the contrast of the different parts of the cell. Then we dehydrate the tissue, embed in hydrophobic resin. And then we do the sectioning, staining. I'll show you later how we do the staining, and then the observation. But this is just conventional EM. I mean, this is just to look at the cells and the content and the subcellular content of the cells, so on. But if you really want to label proteins in your sample, in your cell, to find where these proteins are in the cell, if they're in the ER or in the nucleus or in whatever part of the cell, you're gonna have to do what is called immuno-EM. So you're gonna be able to do some labeling of those proteins for your to localize those proteins in the cell. But then you think that then you're gonna have to use some markers which need to be electron dense. So for your light microscopy, you are using molecules with fluorescence and so on, but because you're using light. But in this case, you're gonna have to use something, a marker that has to be electron dense, so you can actually identify that marker in your organelle or in your cell. Okay, so for this we're gonna use two different methods, which is the pre-embedding method and the post-embedding method. That means that we can do the labeling prior to the embedment in the resin, or after we are doing all this processing. So after you do the processing, you get your section, and then you do the labeling afterwards. So I'm gonna be just focusing on these two procedures right now. Okay, so this is gonna be the next step that we're gonna go through. So how is the pre-embedding method? As I told you, you're gonna have to do a mild fixation. You cannot use too much glutaraldehyde in this process, because then you're gonna fix your proteins, like link your proteins so strongly that they're not gonna be accessible for your antibodies. So this fixation needs to be milder than the one that we used for the conventional EM. Okay. Then we're gonna do our immunocytochemistry. I'm gonna show you how. And then after that, we're gonna do all the rest of the treatment: osmium treatment, dehydration, embedment, blah, 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 like I told you in the beginning. Okay, so this is pre-embedding. You do your immunocytochemistry prior to the embedment, or you can do the other way around. You do also a mild chemical fixation, or even a cryofixation. This is for if you want to. So this is a completely different protocol. I'm not going to be talking about this because this is quite different. But I'm going to be talking you about this one. Okay, so mild fixation, also because we are not going to use too much glutaraldehyde, because otherwise those links are going to be too tight, and they're not going to be accessible to our antibodies. In this case, we are not going to use osmium treatment, because then we're going to darken and fix again too much all these elements in the cells, and we don't want that. We're going to do a mild dehydration, and in this case, look at this. We're going to embed our tissue or our cells in hydrophilic resins. Why? Because we're going to do the immunolabeling afterwards. So we need that the tissue and the resin to be accessible to these water contents. Okay, so we're gonna have antibodies in our buffers or so on. So after we have embedded in hydrophilic resins, we do the sectioning. Okay, the same as I told you before, but we are gonna use different type of grids, which are actually mostly nickel grids, because they are not gonna react with the rest of the reagents that we are using. And we're gonna do the immunocytochemistry, and then on the grids, and then the observation. So to cure these hydrophilic resins, we are not gonna use the oven at 60 degrees, because if we use the oven at 60 degrees to cure these resins, we're gonna destroy our antigens in the tissue. So in this case, we're gonna use this. You see these molds where we have our tissue, and with this resin, but we're gonna use UV polymerization to cure the resin. And actually, this is gonna be in a freezer, so we don't get too high temperature in the tissue, in the block, I mean. So after we have cured this without heating the resin, we get ultra thin sections and then observation. I'm going to show you some of these results using one or another technique: the pre-embedding, the post-embedding, and the pros and cons of using one or another. Okay, so with the pre-embedding, what we're gonna get is a nice subcellular localization of proteins, because we're gonna be able to localize where those proteins are at the subcellular level. And also, it's going to be useful to characterize these labeled cells. Like, we're going to see which kind of cell we are looking at, if it's a neuron or an astrocyte, or I don't know, maybe an axon or something like that. Okay, but in this case, the electron-dense substances that we are using for the pre-embedding method can be two of them. Can be colloidal gold, which are particles of a known size. I'm going to show you a picture of this. Okay, so for example, this is a cell, and here you can see some dark particles, which are actually gold particles, and these, the ones that I'm using, are of a particular size. You can choose different sizes, okay? Or you can use the diaminobencidine, which is a brown stain, which is going to stain the entire cell. So it's going to precipitate onto the cell. And so both of them have, of course, pros and cons. So the pros, when you're using pre-embedding, you're going to have a good preservation and staining of membranes compared to the other one that I'm going to show you later, okay? So because you can see actually the membranes very well defined, and actually you can localize your labeling in organelles or in cells, in the entire cell, okay? But the cons is that you're going to have difficulties to perform labeling of different proteins in the same samples. And I'm going to show you later some examples of this, okay? Because there are some difficulties using two different antibodies and so on. And the post-embedding method also, of course, has some pros and cons. So the pros are that you can actually localize protein subcellular localization. Here I'm showing you, for example, some mitochondria. You can even discriminate if the labeling is in the inner membrane or the outer membrane. So it's kind of cool. Or also you can characterize the cells you are labeling. In this case, the method that I prefer the most, or the most useful, or most people use, is the colloidal gold that I told you earlier on. Okay, so it's you can use particles of a known size and even distinct different particles. So the pros of the post-embedding method is that you have the possibility of performing double labeling. Why? Because you are using your grid to do the immunolabeling on the grid. So you're just treating as you're doing the same as when you are doing immunolabeling on a slide, okay, on a glass slide. You can use different antibodies the same. In this case, you can combine different sizes of particles for each antibody. I'm wanted to show you here, like depending, these are the different concentrations of colloidal gold and different size particles of the colloidal gold. So you can combine which antibody is recognizing which, okay? So you can use antibodies and different antibodies in different sections or even in the same section, okay? But the cons of this post-embedding, as we are not using osmium tetroxide, remember that it's one of the metals that is giving us the contrast of the tissue, is that we're gonna have a poor membrane definition. So you really need to make sure that you are not looking for something that is in, for example, or you need to distinguish very well the membrane of different parts of the cell, so on, because it's gonna be a little bit tricky or a little bit more complicated to get this contrast as well as with the pre-embedding method, okay? And then without the osmium tetroxide or with the osmium tetroxide, I told you that the osmium is crucial to get a good contrast of the tissue, as you can see here, with and without. But since it is a strong fixative and also a staining, if you use them for post-embedding, is prior to your, so you're using osmium prior to your immunolabeling, then you won't be able to really visualize the labeling that you are looking for when you are doing this kind of labeling on your grids, okay? So we can talk about this later if you want. So, and I told you in the beginning, and after that, after all this, you are gonna have to do some staining, which is actually in your grids, okay? Before looking those grids in the EM. This is similar to when you are staining in some sections, like cryostat section or even paraffin sections on your glass slides. Okay, so you're counter staining so you can see something else. So this is something similar. It is called counter staining because you're not using colors, but you're using more metal, hard metals. Okay, so in this case we are using uranyl acetate and lead nitrate, please, sorry, which is a staining for conventional EM. This is actually giving you more contrast than only the osmium tetroxide. So it's as I told you, it's a counter staining. It's helping me to really visualize the darker and the lighter zones in your sample, okay, in your section. But the counter staining is not very much used for immunostain sections, not for pre-embedding or post-embedding, because as you see here, this is on the A is without the counter staining, and in B it is with the counter staining, because there are high probabilities of masking your labeling. Because if you get more contrast with these metals, it's going to be, as you see here, more difficult to analyze or visualize your labeling. Even though, I mean, here you can see some of them, but there are high probabilities that you mask almost all your gold particles, for example. Okay. So, and to finish, I told you that I wanted to show you some of the more specific applications and some of the possibilities in neurobiology, using some of the examples that some of the papers and some of the projects I've been involved in during these years. So maybe they can help you to, like open your mind and see if you can apply one or another technique to your needs, like adjust those techniques to what you're looking for and to your needs. Okay. So actually, this picture is of a neuron, and you can see this the rough ER, mitochondria. This is a nucleolus, so on. Okay. Okay. So some of the things that you can do is that you can have a good correlation of the samples in light microscopy and electron microscopy, because as you saw earlier, you have a block of resin, and then you get ultra-thin sections. But prior to those ultra-thin sections, you can get what is called semi-thin sections that you can stain just with a regular staining. Here is toluene in blue, and you can use them to check if you are actually in the right place that they want to find your cells, or to find your samples, or to find whatever you're looking for. Okay, so you can. This is like a check in light microscopy that you can have just to correlate with what you have in the electron microscope. For example, here is a microglial cell which is actually engulfing some of the cells which are dying in a retina that I did some damage on. And actually, you can see some apoptotic cells which are being engulfed by these microglia, and very nice, like you can see the processes engulfing these apoptotic cells. So you can actually correlate the picture in the light microscopy with the picture in the electron microscope. So you can have a double check of what you are looking for or looking at. Okay. So this is another example. This is actually also conventional EM. I'm going to be telling you here like if it's conventional EM or if it's immuno-EM or so on. Okay. So this is another paper in which we checked the cellular signs in a neurodegenerative disease, and actually in a model of Alzheimer's disease. and we looked for axonal transport. Like there was some defects in the axonal transport in this animal. As you see here in the light microscopy, we see this kind of blobs, and we didn't really know what they contained. And if you look at the EM level at these particular places, what we saw is that we can actually characterize the content of those blobs, that they are like balls. And actually we saw that they were like microtubules and some vesicles and so on. So actually, as before, you can correlate the light microscopy facts or findings with the electron microscope. So you can actually characterize which inside those balls that we were seeing in this animal model. Okay. So another one is an AMD model. AMD is a retinal disease, age-related macular degeneration. And actually, we wanted to characterize this experimental model, and we wanted to know if it was a good model for AMD. And actually, in some of the findings that we got in the retina is that they were accumulation of lipofuncin and so on. But I mean, at this level, you can find some things, or you can describe some of these defects. But actually, the EM level can tell you a lot more. So when we look at the EM level, we saw actually the lipofuncin accumulations in a types of cell, which is the RPE cells of the retina, and also some defects which actually correlate with what we saw at the light microscopy level. So actually, it's a good way of confirming some of the results that you can get at the light microscopy level. Okay. And this is another example of another mutant for retinal disease. This is just to compare what we saw in the fundus of the eye with the findings at the EM level. And actually, we saw also some lipofuncin accumulations and some defects in the photoreceptor cells. As I told you earlier, this is a good correlation model. This is just and most of the times, these kind of experiments can help you to just describe a specific thing that is going on in a specific cell or in a specific organ or organelle. Okay. This is also another example of cellular alterations in an animal model. Was actually an animal without ApoD, and we were looking at defects in the myelin content. And actually, what we saw, there was an abnormal. This is a very good model, also. I mean, a good way of measuring things at the EM level, because actually the myelin, you know, that it has a very periodic, like ROS. And at this point, you can actually measure the periodicity of these lines in the myelin at the EM level. So we detected these defects in the ApoD knockout model. So you do a characterization of the myelin in this animal model. Another example, this is also conventional EM, and it's more or less the same. There was a mouse model without myosin 5A in the retina, and there were some defects in the retina, so it didn't work properly. But actually, the layers of photoreceptor cells didn't look very much altered. And we looked at the EM level in the synapses. And this is another way of characterizing what is going on if those alterations are kind of subtle and they don't really, you don't really see them at the light microscopy level. You can actually try to find something at the EM level. And here you see that there were some defects in the synaptic connections. This is a normal synapse, and we found some defects in the synaptic connections in this animal model. I'm going to go a little bit quicker now. Also some conventional EM, another diagnosis of subcellular modifications in another animal model. This is the same, something similar, but with a different animal model. We found some defects in the synaptic terminals of photoreceptor cells in this animal, and actually you can quantify not only the synaptic terminal, but also which kind of defects you can find at the EM level. And then you can combine conventional EM and immunolabeling. In this animal, there was a mutation in a protein, which is a myosin, and in the retina of these animals, in the RPE cells. And when this protein was not working properly, as you see, the melanosomes were not entering in the area they should be going. Okay, so there were defects in the movement of the melanosomes. So at the immuno-EM level, as you see here, we labeled where the myosin 7A is in the melanosomes, and we found that there was in the actually only in the membrane of certain melanosomes. We can do immuno-EM also in this type of cells. You can do immuno-EM or conventional EM in isolated organelles. Here in this case, for example, we label these beads with antibodies and see which kind of organelles were attaching to these beads depending on the different mutations and so on. And actually you can characterize the type of, for example, vesicles or organelles, mitochondria, whatever. You can do that at the EM level and actually identify the isolated organelles this way. You can do here this post-embedding also with a protein which is in the which mutation causes very severe disease, which is retinitis pigmentosa, and we actually found that this protein is very specifically localized only in the connecting cilium, as you see in the photoreceptor cell. So you can localize subcellularly some proteins. And you can also do this not only with tissue, but also in this case with cell cultures. And this actually was the first picture I showed you with the colloidal gold, the one in B. And this we did pre-embedding in the tissue, in the cells in culture. And we labeled, in this case, it was albumin, and we detected how the albumin is endocytosed in these cells and internalized in the cells. So I mean, you can actually see very well where this protein is engulfed and in which type of vesicles are engulfed in this particle. Okay, and I'm finishing. And this is another example of immunolabeling. In this case, it's post-embedding. We isolated mitochondria. I already showed you one of these pictures before. And if you do immunolabeling, this is post-embedding. Once you isolate the mitochondria, embed them, and then section, you can do immunolabeling on those. And actually, we detected differences in the localization of proteins in the inner membrane and the outer membrane of these mitochondria of different animals, actually mutated animals. And also, this is also another immunolabeling. This is pre-embedding with DAB stain. We could subcellularly localize the proteins, and we characterize. These are, for example, here you see the darkest part is where the DAB was deposited, and you can actually characterize in which type of cells you are getting that labeling. Okay, I'm going to go a little bit faster so it's not too boring. I think this is the last one. And this is another experiment. What we did was this is actually pretty cool because it was hard to get the sections off. We detected here which protein is actually the component of the tip link of the cochlea, of the cochlear cells, of the hair cell stereocilia. And we found, it was previously found, or at least pointed that probably it was cadherin 23, the protein was forming these tip links in the hair cells. But actually, you have to confirm that findings through EM, and it was actually pretty cool. It was hard, but it was pretty cool because I could get these sections of the stereocilia, which are actually really, really, really tiny. But we got this very nice staining of cadherin 23 just or only in the tip link. So this was actually pretty cool and pretty cool finding. And this is just to finish. I've been talking about the processing of samples and tissues for conventional and immunostaining for EM for just regular microscopes. But lately, there have been some improvements in the equipment regarding the electron microscope, because since the early 1950s, The only thing that hasn't changed is the software or some things like that. But right now they have this what is called the serial block phase imaging scanning EM that is gonna be improving all this long processing that I've been talking about. And let me show you how it works, because it's actually an scanning EM, but in block. It's called in block. So how it works, I think it probably, you go. So you have a block, and then you get that one section, and then you see the beam of the electron microscope is like searching, like going through, scanning through the surface of the block, and it's going to get a picture of the surface. So you can continue section and section and section, and then you're going to get a 3D reconstruction of all the pictures of the surfaces that this scanning microscope have taken, but at the transmission electron microscope level. And so you see here, and I have another example of this. Yeah, the next one, you can see the 3D reconstruction I'm talking about here. This is a movie. See, so it's like a 3D reconstruction of all the serial sections that have been going through. Well, I mean, the resolution is not as good as with the transmission electron microscope, but this one has more advantages if you want to, like check an entire block, for example, do like this, all the reconstruction of I don't know, like synapses or like here dendrites or things like that. So you can actually do a 3D reconstruction of the entire block. Okay, so it's actually pretty cool. Even though you don't get a very nice resolution, you can get a good point of the entire block, like a good view of the entire block. And thank you very much. I hope I made myself clear and you can understand everything I went through.